Hello, thank you all for having me. Today I'm going to talk to you about identifying alternative splicing of tumor-specific variants using long-read RNA sequencing. So our current paradigm in cancer is generally to sequence many patients, identify commonly mutated genes, and then develop drugs that target those genes. And in general, this has been a really successful paradigm, and there's been a lot of advances in cancer medicine over the past 50 years. But there's still the challenge of drug resistance. For instance, most patients develop resistance to EGFR tyrosine kinase inhibitors within 9 to 18 months. And there's a lot of different mechanisms of drug resistance. However, a lot of them are both genetic and inherent to the tumor, or they develop over time and are detectable on a genetic level. So for instance, the original driver can develop additional mutations or alterations that allow it to escape the drug. Or it could have a change in expression or amplification that allows it to escape the drug. There's also interactions between different genes that modulate function and allow drug escape. And so I'm going to switch gears a little bit and introduce you all to a Buddhist parable, which is the Buddhist parable of the blind men and the elephant. If you have many blind men around an elephant, each one touches a different part and thinks he's seeing a different thing. So one man may think he's seeing a rope, while another thinks he's seeing a wall, and another a spear. And so I would argue that this is the paradigm by which we look at alterations in cancer genes using short reads. Each assay allows us to detect one part of the puzzle, but doesn't allow us to see the whole elephant. And so with long-read sequencing, we can have a more complete picture of cancer gene alterations and see the whole elephant. And just to highlight why this is important, I want to give one example about somatic mutation and splicing. Or even germline mutation and splicing. And so for this, with short-read sequencing, if we have a gene that has both an alteration in splicing as well as a single nucleotide variant, we can probably detect all the different splicing changes, and we can detect the single nucleotide variant from the short reads, but we can't link them together. We can't understand whether the variant is being affected by the alternative splicing. But with long-read sequencing, we're able to sequence the whole transcript and link together the changes in gene processing and variants. And so my lab developed a tool originally called FLAIR, which was designed to start by assembling isoforms from long reads. And just to really quickly let you know how that works, with long-read RNA sequencing, we get a lot of reads for a gene, but the alignments, are somewhat messy. The ends are messy. You have some maybe misalignments around splice sites. As the data quality gets better, this becomes somewhat less of an issue, but you still have to accurately cluster them into isoforms to be able to understand the underlying biological molecules. So FLAIR clusters them into isoforms and then allows you to quantify them. We next developed FLAIR 2, which improved our ability to detect splicing, as well as incorporating RNA editing. But today I'm going to talk to you about FLAIR 3, which really starts to do this incorporation of all these different types of mutations that could contribute to cancer and seeing them in a holistic manner. And the beautiful thing about this, about incorporating all these different changes, is it allows you to do more accurate functional interpretation of what impact a given variant may be having in a cell. I want to step back a little bit, though, from this grand promise and talk a little bit about quality sequencing data, because this is a type of analysis that has only recently really become possible due to advances in the sequencing technology. So I'm going to show some data from a consortium effort I was involved in called LRGASP, which was for long-read RNA sequencing methods. And here we have a comparison of the number of different combinations of library prep methods and sequencing platforms. The ones highlighted with the red arrows are cDNA PacBio and R2C2 ONT, which are two methods that I used in the data presented in this talk. And then the black arrow for comparison is cDNA ONT. And so in the middle panel, you can really quickly see that the accuracy of both the PacBio and the R2C2 ONT reads is higher. And this is, of course, important if we want to be able to accurately call variants from this data. However, there's other differences as well. On the left, you'll see that the PacBio and R2C2 ONT data have longer average read lengths, but they also have less reads in general for spending the same amount of money, essentially. Moving forward with this, right, what can you detect from these different qualities of data? And I know this is a complex plot, but more yellow, more better. So on top, we can see that even with less reads, but with higher quality sequencing data, we can accurately characterize transcripts with that data, as compared to essentially 10x the amount of coverage with the ONT cDNA, but with lower quality reads. And you might start asking yourselves, why? Why does this matter so much? What is it about like the R2C2 ONT and the PacBio cDNA that is allowing us to do this accurate transcript identification? And one of our main hypotheses is that it has to do with the link distribution. This is showing the coverage across long transcripts. And you can see that although all methods have some bias towards 3' fragments, there's a much more dramatic bias towards 5' fragments in the ONT cDNA. And kind of the more of these short fragmented things that you have present in your sequencing data, the harder it is to really accurately identify a compact set of transcripts that are reflective of the biological reality. And so FLAIR 3 leverages the long reads to identify not just isoforms, but it also does fusion detection. First off, on the isoforms part, I want to just pull up a little bit more data from the LR-GASP consortium, this time comparing bioinformatic tools. So FLAIR is a top-performing tool on multiple benchmarks that balances accurate, annotated, and novel transcript detection. On the fusion side, this is something new that I've developed. And you can see that on a standard benchmark developed by Haas et al. Quite a few years ago, the new FLAIR 3 fusion detection performs comparably to other tools in the field. But if we could compare comparably, like, why develop a new fusion color? But the reason that I wanted to develop this is because there are specific things that other RNA-based fusion collars struggle with. Specifically, fusions with non-genic regions. So if you, say, have a gene at the 5' end, and then the 3' end aligns to a non-genic region, that can just as effectively, say, knock out a tumor suppressor. But it's something that many tools often miss. Also, of course, splicing is my main thing, and so FLAIR 3 works to accurately detect fusion isoforms. CTAT-LR fusion also reports fusion isoforms, but it does not have a lot of filtering, and so its precision is very low. But FLAIR balances the recall and precision pretty well. But the thing that I'm going to focus on the most in this talk today is the identification of variant-aware isoforms. So once you have this complete transcriptome that includes fusion isoforms as well as isoforms of non-fusion genes, we can then identify variants on those isoforms and match them together and quantify how the variant is being spliced. And just really quickly for variant identification, we do this with transcriptomic alignment, which tends to, across multiple different variant collars, improve precision. And by matching these variants to these isoforms, we can do really accurate functional prediction of the proteins that are being produced by this transcriptome. And so today I'm going to talk to you about the application of this to two different cancer studies, one in osteosarcoma, which is a collaboration with the Sweet Cordero Lab, and one in lung cancer, which is a collaboration with the Berger Lab. So first off, osteosarcoma is a bone cancer that is primarily driven by structural variants. In the Circos plots here, the purple lines are all bits of the genome that are not normally connected, that are connected. So there's just massive amounts of structural rearrangements in these osteosarcomas. And for this study, we used osteosarcoma patient-derived cell lines that had been previously sequenced with short-read whole genome sequencing as well as short-read RNA sequencing. And they had also had putative drivers previously identified. So on the right, you can see a table. One of the cell lines, OS052, had P10 loss as a driver. And so what we did is we used R2C2 plus nanopore to get reads with a very high accuracy and asked what new alterations in druggable cancer genes can we identify through long-read RNA sequencing. And so first off, I won't go too into this today, but I think it is really important. We can identify additional information about known drivers, right? So in OS457, the driver was CCNE1 amplification. And from the long reads, we were not only able to detect that, we were also able to detect that CCNE1 had a fusion in the 3' UTR. And this allows us to better understand both the regulation of this gene as well as the mechanism of amplification, because we're also able to show that only one haplotype is amplified. Also, for MYC amplification in OS742, we were able to identify that the amplification correlates with alternative splicing of MYC that causes a distinct protein product. We're also able to identify some novel drivers. So we identified a novel TP53 fusion in one cell line, as well as a KEAP1 deletion. There were other novel drivers, but these are the ones I'm going to talk about today. So in our control cell line, first off, I want to show you that TP53 is expressed and that it produces kind of what we expect to be the normal transcript. But when we get to the cancer samples, we start seeing expression of completely different transcripts, fusion transcripts. So in OS052, we have a fusion of TP53 with MYH10. But then in OS457, we have a fusion of TP53 with a non-genic locus, which is something I mentioned earlier as being a particular strong point of FLAIR3. So here we're able to detect this purely with the long reads, and we're also able to identify two different apparent fusion breakpoints based off of alternative splicing of this fusion. However, both of these fusions probably knock out TP53. Another really interesting finding from this work was a 24 base pair deletion in the KEAP1 gene. KEAP1 is a repressor of NRF2, and overactivation of the NRF2 pathway is known to increase resistance to treatment. So here we can see that in OS742 sample, 50% of the expression of KEAP1 contains this 24 base pair deletion, which leads to an in-frame deletion of 8 amino acids in this protein. And I think some other people at this session have talked about how these mid-sized indels are something that are kind of exclusively detected by long reads. So here's a direct comparison of the long read to the short read RNA. And you can see that the short reads just fail to align around the locus, while the long reads are able to identify this alteration in the cancer gene. Next, I want to talk to you about our work in lung cancer. And today I'm going to particularly talk to you about two patients, patient 1633 and 1427. And this sequencing was done with PacBio Iso-Seq, and we're moving forward with PacBio Iso-Seq for the remaining cohort of 60 tumor and tumor-adjacent normal samples. Now, these patients each had clinical diagnostics run. So one had a BRAF V600E driver, while the other had no known driver. And so First off, I just want to show you this oncoprint pretty quickly. So the solid boxes indicate a alteration driven by a variant. So on the left, in the oncogene side, we can see that we recover the BRAF V600E variant as the only variant in patient 1633. While for 1427, we are able to identify 3 variants in tumor suppressors, which I will get back to. However, for patient 1633, first off, it's great that we can detect this BRAF V600E mutation at all, and that we detect mutations with such precision. We can also identify gene expression in BRAF. And I just want to pause here because this is the limit to the knowledge you would get with short reads, right? You could do an assay for gene expression. You can do an assay for the variant. Great, that's the information you have. But with the long reads, we can move forward and start looking at, first off, how is the splicing changing? So we can see that the orange BRAF 220 isoform has significantly increased usage in the tumor sample as compared to the matched normal. But then, when we do the isoform variant matching, it gets even more interesting. And before I show you the data, let's just take a look at what isoforms overlap this variant, right? So the blue and orange isoforms both overlap this variant. So intuitively, we would expect it to be expressed on both of those isoforms. However, that is not actually the case. We see that the BRAF V600E variant is only expressed on the BRAF 220 isoform. Which is really cool and allows us to do much better functional prediction of the impact this isoform is this variant is having. Interestingly enough, the BRAF 220 isoform is the canonical isoform for this gene, and so it probably is the one that is targeted by BRAF inhibitors. So this is a patient that you could pretty confidently recommend treatment by a BRAF inhibitor. For the other patient with no known driver, we were able to identify 3 alterations, one in KEAP1, one in TP53, and one in CDKN2A. The KEAP1 variant behaves the most like you would expect a variant intuitively to behave. So there's 2 isoforms of KEAP1. The variant is expressed equally on both for heterozygous expression. However, TP53 and CDKN2A are a bit more complicated. So for TP53, I'm going to focus primarily on the primary isoform that should produce the functional protein for TP53. And for this, we see 3 different distinct alleles. So in the normal sample, the patient is heterozygous with both the reference allele and a benign variant. But in the tumor sample, we see the reference allele, the benign variant, as well as a biased expression towards the damaging variant. And this likely is an example of potential somatic variation in our cells, so the damaging variant is not in all of the cells that were sequenced in this bulk sample. For CDKN2A, we also see a bias towards the mutant allele, although it's a complete bias. We have a complete switching in both isoforms from the wild-type allele in the normal sample to the mutant allele in the tumor sample. And what's likely happening here is that one allele of CDKN2A is getting deleted, while the other has a variant. However, we can also identify this change in splicing, right? This increased usage of the CDKN2A 214 isoform. Now, CDKN2A is a really interesting gene because its 2 isoforms that shown here produce 2 completely distinct proteins. So the CDKN2A 201 isoform, which is the more canonical one, produces the P16 protein, which functions through the CDK4 pathway, while the CDKN2A 214 isoform produces the P14 protein, which functions through the TP53 pathway. And so these distinct proteins are having distinct functions, but they're also being affected differently by this variant. So the red arrow on this plot is showing the position of our particular variant. And the variant is right in the middle of a functional domain on CDKN2A 201. And so, this is where the prediction of it being a damaging variant comes from. However, the variant impact prediction does not generally take into account secondary isoforms. So on the CDKN2A 214 isoform, it's right at the very tail end of this protein, and so may not actually be knocking out the function of the CDKN2A 214 isoform, which functions through that TP53 dependent pathway. And so, this is just one example of how this interaction of variants and splicing can really enrich in our understanding of cancer gene alterations and how they may be impacting the patient. So going back to the oncoprint, I just hope I've shown you that the long-read RNA analysis recovers both drivers and provides additional insights into variant function. And then again, back to our parable, this long-read sequencing allows us to just look at all of these alterations together and see the whole elephant, which I think is going to be very powerful. So with that, I would love to thank both my lab as well as my collaborators at Fred Hutch and UCSF. Allison Tang and Cameron Soulette were the original developers of FLAIR, and I'm just building on their amazing work. Thank you very much.